Love the little countdown for the video. All right. So last week, we were learning about the cell cycle, and we talked about how the cell cycle is controlled and regulated by all of these signals that cells get, both from outside of the cell and inside the cell, as they check on what's going on, and they're making sure that all the DNA is copied and that they're ready to go on to the next phase and have enough nutrients and all that stuff. And if that's true, then we talked about how there are these different groups of proteins, the cyclins and kinases, that tell cells, they kind of work together to tell cells when it's okay to move past those checkpoints. Those proteins that control the cell cycle obviously are coded for by genes. And so part of what happens in that process is if the cell is receiving signals from outside the cell that tell it that it needs more of those molecules that say go ahead and go, it activates genes that produce more of those cyclins and kinases in the cell. And so as their concentration increases, more of the kinases bump into the cyclins and activate them, and that starts to tell the cell to move forward. So today we're going to kind of back up a step, and we're going to talk about, in general, how the expression of genes in our DNA is regulated and controlled. How do your cells know which genes to read and make those proteins or make those RNA molecules, and which ones are not important for that particular cell? So this does connect back to regulating the cell cycle and the process of development and the other things we're talking about in this unit. But this is also kind of a broader topic because this is really how cells regulate all of their genes and know which ones to turn on and which ones to turn off at any given time. So we'll talk a little more about why that's important as we go, but that's kind of the topic for today. So first we have to go back to a question that I think we probably started talking about in the last unit. We talked a little bit about how the definition of a gene has kind of evolved over time. First, Mendel just knew it was responsible for a trait, and then they figured out that a gene was found on a chromosome. And then eventually they thought maybe one gene coded for one polypeptide. But it needs to be a little more comprehensive than that, because as we've talked about, there are genes that do all of that, but around that, there's like the promoter region with the tata part that tells something to begin transcription, and there's the end of the sequence, and those aren't actually part of what makes the protein. So this definition needs to be a little more inclusive. So we have to consider the parts of the genes. Remember, the introns are the pieces that are cut out, that are in between. They're still in there. They're still transcribed, but they're not translated. So we have to include those. We have to include the promoters and other things like that that are part of that gene. They're within that region, but they don't actually end up being part of the polypeptide. So just saying that a gene is equal to one polypeptide, or it's the instructions for one polypeptide, isn't quite accurate and complete. We also have to keep in mind that not every single gene codes for a polypeptide. Some of them, think about all the different types of RNA that we talked about in the process of transcription. Obviously, messenger RNA, or in the process of protein synthesis. Obviously, messenger RNA is made directly off of DNA, but we also talked about the fact that ribosomes are partly made of RNA, and there's transfer RNAs that just float around your cell and grab amino acids and bring them in, right? Those other types of RNA, the RNA in the ribosome, and those transfer RNAs are also made the same way messenger RNA is. The only difference is that, like a transfer RNA, it's transcribed like a messenger RNA, but then instead of going out to a ribosome to be translated, it just folds up into that little like clover leaf. I'm not going to be able to draw that real well. You remember that shape? Something like that. It just folds up like that, and then it floats around the cell just as this strand of RNA, and it grabs amino acids and brings them into ribosomes and goes back out and does that. But this RNA was still copied based on a DNA code in the nucleus. Does that make sense? So not all DNA codes for polypeptides at all. Some of it just codes for RNAs that have their own job in the cell. So a complete definition of what a gene is, and kind of the most modern definition, is something more like this. It's a region of DNA that can be expressed to produce a final functional product. It might be a polypeptide. It might be an RNA molecule that goes out and has some function in the cell. So today we're going to talk about this process of how we get from a gene to a final product. Could be a polypeptide, it might not. And the different places along that pathway that your cell can either kind of dial up this process so that there's more transcription and more translation and more of that product being produced, or dial it down or turn it all the way off to produce less of that type of RNA or of that protein. Okay, so that's what we mean when we talk about controlling gene expression. It's cells being able to kind of turn on and turn off different parts of the DNA to control how much of each polypeptide or each type of RNA they make in the cell. So before we get into these different parts of control, I want you to talk about a couple different questions here. I'm going to put the first couple up here. Actually, I'll put all three of them up here. So I want you to talk through this. How does the genetic material of two daughter cells at the end of mitosis compare to each other? Your body was formed through many mitotic divisions. That means mitosis happened lots and lots, right? You were originally formed as one cell that went through mitosis again and again and again and again to form you. So how does the DNA of all your cells compare? And if you're the result of tons and tons of mitosis, why aren't all of your cells identical? Turn and talk about those three things for a minute.
Yeah, I think the DNA is the same. But the way the cell uses the DNA makes the same. All the DNA is here. Or makes all your cells usually the brain. It needs to like change. Like I feel it's like an evolution kind of thing, you know? Like a change. I don't know. Yeah. Maybe like function. Yeah, I don't know.
All right. So let's logic through this quick. So based on what you know about the process of mitosis, which we studied last week, how does the DNA of the two cells you get at the end compare to each other and to the one you started with? They should all be identical, right? Barring the fact that, yes, we know in real life there are occasional mutations and mistakes that are made and get through, but if the process all goes as planned, they should be identical. You were formed because of mitosis happening over and over and over and over. So what's true about the DNA in every single one of your cells? It should all be identical. Are all of the cells in your body identical in how they function and the proteins they create and all that kind of stuff? No, your brain cells, your blood cells, your heart cells, your stomach cells, all very different in terms of how they use the instructions that they're given. Okay, think of your DNA like just this humongous instruction book for every single part of your body. Your stomach doesn't need to read about what your brain does. It only needs to read the section on how to be a stomach cell. It only needs to make those proteins and use those genes. So this process of controlling and regulating when genes are turned on and off is really important to how your body functions and how your brain cells are able to act like brain cells instead of acting like stomach cells. So that's why these types of controls and regulation are really important, is that's what makes all the different tissues and all the different cells of your body. So, this process is of regulating gene expression has to do with turning genes on and off. Again, there are both internal and external signals that are involved here. And that idea of cell signaling comes into play here as well, because sometimes the signals that tell your cells to turn on a gene and start making a certain protein come from outside the cell. It might be those growth factors that tell them to turn on proteins to stimulate cell division. It could be hormones that's telling your cells to start producing something else to trigger other developmental changes. And like we just said, this is kind of the sciencey way to say what I was just saying about brain and stomach and all that kind of stuff. This is necessary for cell specialization. Specialization means only producing certain types of proteins for its particular job. So here's the sciencey language for saying what you just said. All the cells in your body have genomic equivalents. The genome, the code of DNA, the A's, T's, C's, and G's that you have in every single cell is essentially identical. Like we said, barring the occasional mutation that, yes, does happen, but they all should be identical. But they have differential gene expression. So they have the same DNA, but they use it differently. They express or make polypeptides and RNAs based on different pieces of the DNA in different cells. So obviously the main place that everything can be regulated is with the DNA itself, right? By actually either encouraging transcription of certain parts of the DNA or blocking transcription of certain parts of the DNA. So most of the regulation that we talk about will be right at the very beginning of transcription, but there are actually several other places along the way that regulation can occur, because this isn't always an all or nothing. You may want a gene to be turned on, but only to make a little bit of that polypeptide for that cell, whereas a different cell might need it turned on and make a lot of that polypeptide. So there are a lot of places kind of along that transcription and translation pathway that this regulation happens. But like I said, most of it starts at the beginning of transcription with the actual gene, the actual piece of DNA itself, and whether it's going to be read or not. So you may remember seeing pictures like this before. This part should look familiar to you, right? When we learned about protein synthesis, you know that a gene codes for a polypeptide. So that's the code, the A's, T's, C's, and G's, that will actually be transcribed into a messenger RNA and then made into a polypeptide at a ribosome. And we talked about how at the beginning of a gene, there's this promoter sequence that often includes that TATA sequence. And that's where the transcription initiation factors kind of bind on and help RNA polymerase to find its spot. So we talked about these general transcription factors. They bind to the promoter here, and they help RNA polymerase down here to find its spot and get started. But in these diagrams, you'll notice there are some other pieces of the DNA that help with this as well. Upstream, which means kind of backwards before the promoter, because remember, transcription happens this way, so upstream means go in the other direction. There are a few pieces of the DNA here called control elements. Those are bits of DNA that don't actually code for a polypeptide, but you can think of them kind of like part of the genetic switches that turn this gene on and off. So there are proteins that are called specific transcription factors. Specific means that they're part of they're part of the switches instead of actually activating the gene itself. They bind to the DNA at these control elements. So this right here is a specific transcription factor. It's a protein that binds here, and you can see what those specific transcription factors do is they help these other proteins to get to where they need to go. They help this whole initiation complex to form more easily. There's another control element that's shown in here. This DNA bending protein also recognizes a sequence of DNA. It's not really labeled up here, but it bonds there and literally helps curve that molecule back around. So the control elements are right next to the promoter, again, to help this all form more easily. In this diagram, these are labeled as activators. And like I said, they're helping these to bond and to get in place. So activators kind of turn on the gene. There can also be repressor control or transcription factors that inhibit or stop transcription from happening. So a repressor, if there was a different protein that bonded up here, they would still bond to these control elements, to some piece of the DNA, but instead a repressor would turn off transcription. They would make it harder for all these other proteins to bond and for RNA polymerase to find its spot so that it would not be able to transcribe that gene as easily. So a lot of the control of which genes are turned on and off has to do with what combination of these activator proteins and repressor proteins are made within a cell, because those activators and repressors then go to bind to the DNA and either turn on the gene that they regulate or turn it off. Any questions about that? Okay, sometimes your body wants to be able to turn on a lot of things at once. And so that is called coordinate control. If there are multiple genes within a cell that maybe all work together to produce enzymes for a certain chemical pathway, or you need a few polypeptides to come together to build a functional protein, sometimes there are multiple genes that need to be turned on at once. And so each of the genes has its own set of specific transcription factors. And the ones that are made in that cell will determine what's turned on and off. If we need to turn on a lot of genes at the same time, they each have their control elements, but if they all need to turn on at the same time, then the activator protein is a protein that's able to bind to all of them. Basically, you can think of it as one switch that controls like all the lights in a room. That activator protein will fit with all of those different control elements. So as soon as the cell has made a bunch of those activator proteins, they go and they bond here and bond here and bond here and turn on this gene and this gene and this gene all at the same time. And so the cell starts activating and transcribing all of those genes to make all of those products at the same time. So these give cells a lot of ways to control what they're making and how much of it they're making. This type of coordinated control is often triggered by external factors like hormones. Like I said, if you think about some of the developmental changes that happen, there are a lot of things that start happening in your body all at once. So those hormones go out to many different cells, and they trigger cell signaling pathways in many different cells, and they tell all of those cells to start making these activator proteins that then go and turn on a whole bunch of genes so that your body starts going through all these different changes and things are happening everywhere all at once. So that's kind of how your body's able to activate a bunch of stuff all at the same time. Questions? Okay, so that all has to do with the very, very beginning, with whether or not transcription of the gene happens in the first place. But there are other places along the pathway between a gene and the finished protein that regulation can happen. So this is just kind of a summary of several other places that this can happen. Because like I said, this isn't just an on-off switch. This is also a way that cells can control how much of a particular product they're making. And so in addition to the beginning of transcription, after transcription happens, remember you have that pre-mRNA molecule That needs to have the end modifications put on, and the introns need to be spliced out, and it needs to be kind of edited to make that finished product. That's the next place that things can change and cells can kind of control what products are being made. Is that idea of alternative RNA splicing, which happens right in here. Depending on which introns are being cut out and how that RNA is edited, the cell can control which products are being produced from that gene. Then that finished messenger RNA has to leave the nucleus and go out to a ribosome in the cytoplasm. And between those two, that's another place that the cell can control how much that protein is made. There are other proteins involved in initiating translation, and whether the messenger RNA can actually bond to a ribosome and the ribosome can start to read it. We're going to talk about a couple of other types of RNA that actually interfere at this point. So you could put a note here, because the next couple things that we're going to talk about kind of work right there. As translation is being started, sometimes there are these random other little RNA molecules that actually kind of get in the way. So translation can't start. Even if translation does start, there are also enzymes in the cell that break down messenger RNA. And so depending on how quickly that happens and how many of those are in the cell, once you break down a messenger RNA, it's not going to be translated anymore. So if a cell needs a lot of a particular polypeptide, then they just don't make the enzymes to break down the messenger RNA. So the messenger RNA just kind of hangs out in the cytoplasm. And it attaches to a ribosome and it gets translated. And then it attaches to another ribosome and gets translated again and again and again. And you can use the same message over and over. But if a cell does not need very much of that protein, it can translate that messenger RNA once or twice and then break apart the messenger RNA and we're done. Does that kind of make sense? So that's another place cells can kind of dial it up and down. Then even after translation's done, there are still those kind of finishing touches that need to be put on the protein, those chemical modifications and how the protein folds up. And sometimes that process, how those proteins are folded and modified, the proteins can be folded and modified in a way that makes them inactive. We've talked in a lot of the signal transduction pathways about how a protein is inactive until it becomes phosphorylated and then it can be turned on. So sometimes depending on how it's modified and folded, the proteins may or may not be active when they're made, depending on what the cell needs. And of course, the proteins themselves can be broken down if it's something the cell doesn't need anymore. So all the way along this pathway, there are opportunities for the cell to control how much transcription is happening, how much translation is happening, and how much of this polypeptide is in that cell. And the reason there are so many different places to control it is because that lets the cells really, really fine-tune exactly what they need for the type of cell that they are, and the conditions, the environment that they're in, and the signals they're receiving. So they can respond and change as the environment around them changes. So we're going to go back and talk a little bit about that initiation of translation part. In recent years, as scientists have studied this stuff more and learned about things like genetic switches and how genes can be controlled and turned on and off and all of that, one of the things that they found is that there are actually, there's more stuff happening in our DNA that we realized. For a long time, scientists just thought that in between the sequences of DNA that code for polypeptides, there were just these big long stretches of junk DNA that didn't do anything. Well, scientists are now figuring out that a lot of that junk DNA actually codes for small RNA molecules that have purposes in the cell. They have a job besides things like tRNA. And there are a few different types. We're going to talk about two mainly today. One of them is a group called microRNAs, or it's abbreviated miRNAs, M-I-RNA. So these are very short little bits of DNA. They might only be, 12, 15 bases long. They're single-stranded. They're coded for by the DNA just like a messenger RNA would be. But what they do is they go out into the cell and they bind to a messenger RNA on a complementary sequence. And because this mRNA has bonded there, the ribosome can't. And the ribosome can't start translating that RNA because this other little bit is in the way. Does that kind of make sense? So like if this is your messenger RNA with the sequence of bases here, if one of these microRNAs comes in, it might only be a few bases long. So this is the mRNA. But if it's blocking the start codon, right? If this down here is my AUG, where the ribosome is supposed to start translating, that first transfer RNA can't come in and bond there because there's already something bonded to it. And so the ribosome can't start translating. So it physically gets in the way and blocks translation from happening. And that can be a permanent thing, and that can then go and just get broken down so it never gets translated, or it can be a temporary thing where then that microRNA kind of detaches and comes off and then later allows it to be translated, okay? There are also these other ones called small interfering RNAs. They're similar, they're small, they're single-stranded RNA molecules, and they can block gene expression through a process called RNA interference. And this is what we're going to watch a short video about. This is about 15 minutes long, and this will be kind of the last thing for today. After this video, if you have any questions about the mitosis lab or anything like that, you'll have a little bit of time to ask about those things. But as we watch this, do you have these? Did I print these questions in your notes? Okay, so these are a couple of things that I want you to just think about and maybe jot down as you're watching this video. Is what was the original function of RNAi? That's what they refer to it as in this video. It stands for RNA interference. Like, why might this have first evolved in the first place? What was its purpose? And how have scientists kind of taken this and used it to try and benefit people? Those of you who are at home, would you rather I just put this link in the chat so that you can go and watch it on your own? Or do you want me to stream it through here? How about this? I'll throw the link in the chat. If you want to go and watch it on your own so you get slightly better sound quality and stuff, you can. If you want to stay here, I'll stream it through the Meet. It's up to you, but please do watch this so you get the last bit of information for today. Okay.